Today let us do pigments, quanta and photons. To understand how light energy is captured by pigments of plants and converted into chemical energy, we will first understand the nature of light. Light is a form of electromagnetic radiation emanating from the sun. So it is a form of electromagnetic radiation. It consists of photons which are produced when the atoms heat up. These photons, they travel as waves, and this is the only form of energy visible to the human eye. No, this is not correct. The wavelength which is visible to the human eye is of 380 nanometers to 750 nanometer, which is known as the visible light spectrum. It is the wavelengths lower than these wavelengths, and there are radiations which are wavelengths which are greater than this and which are also lower than this visible spectrum, but they are not perceived by the human eye. So as per the wavelength, the electromagnetic spectrum is divided as gamma rays, X-rays, UV rays, visible light, infrared, microwave and radio waves. These are all broadly categorized as these different categories depending upon their wavelength, and the visible spectrum is of 380 nanometers to 750 nanometers, which is utilized by green plants. So to define a quantum, the amount of energy contained in a specific wavelength of light over a distance is different from amount of energy contained in every other kind of light over the same distance, and this is called a quantum. Say for example, the quantum of energy of infrared rays will be different from the quantum of energy carried by the UV rays. A measurement or way of defining light is called a photon. Actually, to give a unit to this radiation, the term photon was conceived, and a quantum is defined as the amount of energy each specific wavelength of light contains in a photon. The electromagnetic spectrum, as we know, it includes gamma, UV, X-rays, infrared, microwave and radio waves, and each of these wavelengths, depending upon their wavelength, has a specific amount of energy. As the wavelength increases, the amount of energy carried by it decreases. That means it is inversely proportional. So as we have seen, there are wavelengths which are shorter than the visible spectrum, which means they have higher energy. So is this higher energy wavelength, is it good for us or is it bad for us? Fortunately for us, the upper atmosphere of our planet filters out most of the really high energy light before it reaches us and the plants. That is, wavelengths which have very high energy is broadly filtered out before it reaches down to the surface of the earth. Infrared light, which has greater wavelength than the visible spectrum, has a low energy photon. Infrared light does not contain enough energy for photosynthesis. UV light, which is a shorter wavelength, it has too much energy and in a sense it cannot be controlled by the plants. UV light intercepted by plants can create free radicals which can break chemical bonds in an organism. This is detrimental to the plant. Plants in fact have pigments to protect them from UV light. Visible light, however, seems to be just right for plants to use to move electrons around. So it is only the visible light which is beneficial for us, and the plants have adaptations to withstand the ill effects of UV light, and otherwise the atmosphere also filters out UV light and it is not permitted to reach down into the lower levels of the atmosphere. Pigments are molecules that absorb specific wavelengths, that is specific energies of light, and reflect all others. Electrons in a molecule can exist at specific energy levels. So that means electrons within these pigments, they also can exist at specific energy levels. Normally they exist at the lowest possible energy level that they can reach. However, if enough energy comes along to boost them into the next level, they can absorb it. They can absorb that energy and occupy a higher level. Any light energy that does not have enough or has too much energy cannot be absorbed and is reflected. That means specific pigments can absorb specific lights, that is energy, specific energy levels only can be absorbed by these pigments. So each pigment is specific for a wavelength of light. The electron in the higher energy level, they want to return to its normal energy level, and this can happen in several different ways. The extra energy can be converted into molecular motion and it can be lost as heat. Some of the extra energy can be lost. Some of the extra energy can be lost as heat, while the rest of it is lost as light. This re-emission of light energy is called fluorescence. The energy can be passed on to another molecule. This is called resonance. The energy and the electron can be transferred to another molecule. So we see that the extra energy captured by any electron, it can be either lost as heat, or it can be re-emitted as fluorescence, or it can be passed on to another molecule. Plant pigments usually utilize the last two of these reactions to convert the sun's energy into their own. That is, they pass it on to another molecule, or the electron can be transferred to another molecule. When chlorophyll is isolated from the enzyme it is associated with, the second scenario can be seen to happen. I hope you have understood this. That is when actually chlorophylls, they do not exist in isolation within the plants. They are always associated with proteins. So when they say that chlorophyll is associated from an enzyme, that is, it is when it is associated with the protein, it will capture energy and it will be transferred to by resonance to another molecule. However, when you isolate the chlorophyll from its protein component, the energy absorbed can be lost as light, which is the second scenario, and that is the chlorophyll will fluoresce. So now we try to understand which of these pigments, the pigment absorbing UV light, or pigments absorbing infrared light, or pigments absorbing the visible spectrum, which of these pigments would be the ideal pigment for a chloroplast. A collection of pigments which absorb light in all wavelengths of lights, that will be an ideal choice. That is, the pigments absorb all kinds of lights and it is utilized by the plant, and so a black seems to be a logical choice. However, if plants had pigments which absorbed UV light, X-rays, which are very high energy wavelengths, this would mean that so much energy would be absorbed that electrons would be knocked off. That is, all radiation effects can be seen. Electrons are being knocked off their orbitals and the molecule would be destroyed. If plants absorbed infrared and radio waves, there would not be enough energy for an electron transfer, just enough to warm up the molecule. So this is also not ideal. Pigments that absorb in the visible region, they gain just enough energy to boost an electron to the next level. So this is the most ideal scenario, where pigments, they absorb light in the visible region. This figure shows the absorption spectrum of chlorophyll A and chlorophyll B. As we can see, they are absorbing in the 400 to 500 range, which is the blue range, and the orange red region, which is 640 to 720. Broadly, these wavelengths are captured by chlorophyll A and B. They are two absorption peaks are being formed. So chlorophylls and carotenoids. The upper diagram is of chlorophyll A and the lower one is of beta carotene. Chlorophyll B, this molecule has a structure similar to that of chlorophyll A. It absorbs light of 453 nanometer and 642 nanometer maximally. It is not as abundant as chlorophyll A and it probably evolved later. It helps increase the range of light that a plant can use for energy. Carotenoids are a class of accessory pigments that occur in all photosynthetic organisms. They are completely hydrophobic, fat soluble as can be seen from the carbon chain, and they exist in lipid membranes. Carotenoids absorb light maximally between 460 nanometer and 550 nanometer. They appear red, orange or yellow to us. Carotenoids protect the plant from free radicals. The most important function of carotenoids seems to be protecting the plant from free radicals formed from ultraviolet or other radiation. Free radicals, they are dangerous because they contain an extra odd electron they don't really want to have. This means that they are constantly trying to get rid of this extra electron. They do this by attacking whatever bonds they can. In animal systems, it is speculated that vitamin E plus oxidized radical gives a radical plus vitamin E which is oxidized. Vitamin E oxidized plus carotenoids, vitamin E plus carotenoids oxidized. Carotenoids oxidized plus vitamin C, carotenoids plus vitamin C oxidized. That is, vitamin C is protecting. In animals, the vitamin C can be flushed out of the system since it is soluble in water, which none of the other molecules are. Whether this same series of transfers occurs in plants is unknown. In humans, taking vitamin E or beta carotenes in themselves do not protect against oxidants unless there is a supply of vitamin C to flush it out. In plants, the vacuole is a generally safe aqueous repository. The oxidized C may end up there, whereas carotenoids could not. Xanthophylls is another class of pigments. They are essentially oxidized carotenoids and they contain oxygen. They are usually red and yellow and do not absorb energy as well as the carotenoids. Even xanthophylls are also fat soluble. Anthocyanins are another class of pigments. These pigments are also present in plants. These pigments contain copper and are stored in the vacuole of a plant. Because they, unlike the other fat soluble pigments, they are water soluble. The structure of anthocyanins in the red algae and BGs, how does it differ substantially from the above pigments? Can you take it up as an assignment?